Imagine for a second that you could take a standard everyday photograph of a massive crowd of people.
Okay.
And just by looking at the physical arrangement of that crowd who is standing next to whom, how tightly they're packed together, maybe even the slight tilt of someone's shoulders.
Right, the subtle details.
Exactly.
Yeah.
Just by looking at that, you could instantly know the exact genetic code of every single person in that picture.
I mean, it sounds completely like science fiction. You're talking about inferring invisible foundational biological data strictly from a surface-level physical pattern.
It really does sound wild.
Yeah.
But welcome to the deep dive, because today we are looking at a brand new, groundbreaking preprint paper from Stanford and Yale researchers about an AI model called STORM.
Yes, STORM, Spatial Transcriptomics and Histology Representation Model.
That's the one.
Yeah.
And it is basically doing this exact science fiction thing to human cells. They're teaching an AI to see the invisible molecular secrets of human tissue using really cheap everyday medical images.
Which is huge.
Right. Okay, let's unpack this, because if this works the way the research suggests, it could completely revolutionize how we treat cancer.
Oh, absolutely. The implications here are massive. And, to understand why this matters to you or to anyone who might ever interact with the healthcare system, we have to look at the tools doctors currently rely on.
Setting the stage.
Exactly. The gold standards for diagnosing cancer today are kind of trapped at two extreme ends of a spectrum.
Okay, what do you mean by extreme?
Well, on one end, you have methods that are cheap and universally available, but they are just way too simplistic to reveal the deep molecular drivers of a disease.
Like they just don't give enough detail.
Right. And then on the other end, you have these cutting-edge technologies that show you absolutely everything, but they are far too expensive and complex to use on everyday patients.
So we're stuck.
We are. And STORM acts as the ultimate translator between those two worlds. As we go through the research today, the question you should really keep in the back of your mind is how much hidden information is sitting right in front of our eyes. If only we knew how to look.
I love that.
Yeah.
So let's start with that frustrating gap in modern medicine, the map versus the landscape, basically.
Yeah.
What is the cheap, accessible method that doctors are using right now?
So that would be H&E staining.
H&E.
Yeah, hematoxylin and eosin. If you've ever looked through a microscope in a high school biology class, you've probably seen this.
The pink and purple slide.
Exactly. The pink and purple dye, it's a century-old technique. It allows human pathologists to see the physical shapes of the cells, the morphology.
Okay.
They look at it and ask things like, is the cell enlarged? Is the tissue structure disorganized? Does it look like a tumor? It is literally the absolute bedrock of clinical diagnosis today.
But I mean, it's really just shapes and colors.
Yeah.
Right? It doesn't actually tell you what genetic software is running inside those specific cells.
That is the crucial limitation. For the genetic software, you need the other extreme we talked about, which is spatial transcriptomics. Or ST.
ST, and this is the expensive one.
Very expensive, but it's a revolutionary technology because it doesn't just show you the physical shape of the tissue. It maps the exact gene expression, like the thousands of genes turning on and off directly onto that physical anatomy.
Oh, wow.
Yeah, it reveals the actual molecular machinery at work in its native tissue context.
Okay, so if I'm understanding this, H&E is like looking at a satellite image of a city.
Okay, I like this.
You see the buildings, right? The roads, the general layout. You can tell if a neighborhood looks residential or industrial.
Sure.
But ST, spatial transcriptomics, is like tapping every single phone line in that city to hear the actual conversations happening inside those buildings.
Yes.
So you know exactly what the people are doing and planning, not just where they live.
That captures the difference perfectly, but imagine the actual cost of tapping every single phone line in a major city.
Right. It would be insane.
Exactly. That's why ST is largely stuck in elite research labs. It is insanely costly. It is incredibly complex to run. And it's super low throughput. You just cannot realistically do it for everyone who walks into a local clinic with a suspected tumor.
Which means, unfortunately, doctors are making life or death treatment decisions based mostly on those pink and purple satellite images.
Unfortunately, yes.
Have people tried to use AI to bridge this gap before? Like just having a computer look at the cheap image and guess the expensive data?
They have. Scientists have tried to train AI models to do exactly that. Look at the cheap H&E image and predict the ST data.
Did it work?
Not really. What's fascinating here is that those previous attempts largely failed because they suffered from a kind of tunnel vision.
Tunnel vision?
Yeah, they tried to look at isolated individual spots of tissue, like just a single cell or a tiny cluster, and predict what was happening in a vacuum. They completely lacked an understanding of the surrounding neighborhood.
Oh, I see. Like trying to guess what a business does just by staring super closely at its front door without, like, noticing it's located in the middle of a busy shipping port.
Yes, exactly. They lacked what biologists call spatial context. A cancer cell behaves very differently if it's surrounded by other aggressive cancer cells versus if it's, say, surrounded by attacking immune cells.
That makes sense.
Tissue organizes itself in complex architectures. If an AI doesn't understand the neighborhood, it can't truly understand the biology.
Okay, so you have this huge AI problem where previous models lack spatial context. How did the researchers at Stanford and Yale actually build STORM to fix that?
Well, they started with unprecedented scale.
How big?
They trained STORM on a massive data set of 1.2 million spatially resolved spots.
Whoa, over 1 million.
Over 1 million,
Mhm.
each with matched H&E images spanning 18 different human organs.
18.
Yeah, breast, colon, skin, heart, kidney, lung, brain.
So it's not just a breast cancer model or a lung cancer model.
Exactly. This diversity is critical, so the model learns universal biological rules rather than just memorizing the quirks of one specific organ.
Okay, 1.2 million spots is a huge data set. But how do you actually structure the AI so it doesn't just get tunnel vision like the older models did?
They gave STORM a spatially aware hierarchical design.
What does that mean in plain English?
Basically, step 1, the model looks at a single spot using a spot encoder to grab the local physical and genetic features.
Okay, so it looks at the front door.
Right. But then comes step 2, a spatial encoder. This module doesn't just look at the one spot, it analyzes a 5x5 grid of neighboring spots all at once.
Oh, so it zooms out.
Exactly. It fuses the information while preserving the exact spatial coordinates. It actively learns how the neighbors interact with each other.
So it's looking at the entire neighborhood block. That's clever. But how do you actually teach it? I mean, how do you test an AI on 1.2 million spots to make sure it's actually learning something useful?
They used a brilliant strategy called masked pre-training.
Masked pre-training.
Yeah. The researchers would take this rich, complex data, and they would randomly mask or hide some of the biological information in certain spots.
Just blindfold the AI to certain parts.
Exactly. Then they force the AI to reconstruct that missing data just by looking at the surrounding spots.
Wait, like a game of biological Sudoku,
Yes.
where it has to deduce the missing cells function based on the rows and columns of the cells around it.
That is an excellent way to think about it. And because the AI doesn't know which spots will be masked on any given training run, it cannot just memorize the data set.
Right, wait, let me jump in here because I can play skeptic for a second. You're saying they essentially play molecular fill-in-the-blanks.
Mm-hmm.
How do we know the AI is actually learning biology and not just, memorizing the test answers from those 1.2 million spots?
It's a fair question. But to successfully fill in the blanks across 18 diverse organs, the model is literally forced to learn the underlying grammar of how human tissue organizes itself. It learns the fundamental rules, not just the patterns.
Okay, but I'm still stuck on the core mechanism here.
Mhm.
How does the shape of a cell tell an AI what genes are active? I mean, if it's just looking at pink and purple dye, how does that translate to genetic software?
Think about human biology. If you decide to start a heavy weightlifting routine, your genetic software activates new proteins, right? And over time, your physical body shape changes.
Yeah, you build muscle.
Cells do the exact same thing on a microscopic level. If a cancer cell turns on a gene to aggressively hoard fat for energy, the cell physically changes. It literally swells.
Oh, I see.
Its membrane bulges outward. The nucleus might get pushed to the side. Human eyes just see a blurry pink blob on a slide.
But the AI sees the subtle changes.
Right. STORM, having played millions of rounds of that biological Sudoku, has learned that this specific microscopic bulging, combined with the layout of the neighboring cells, corresponds to that exact fat-hoarding gene.
Okay, that makes total sense. The genetic software changes the physical hardware. All right, so you have an AI that understands the grammar of how tissues organize.
Yeah.
But a smart AI in a lab is kind of useless unless it can spot something human doctors are missing in the real world.
Yeah.
Did they actually test this on a real human tumor?
They absolutely did. They used STORM for what they call spatial domain discovery.
Spatial domain discovery.
Which basically means asking the AI to map out the different microneighborhoods inside a tissue sample. And they provided a fascinating example in the paper involving a kidney cancer sample.
Right, the KC2 sample.
Mm-hmm.
This part of the research was totally wild. So if I remember correctly, a highly trained human pathologist looks at this pink and purple slide and draws a circle around a massive chunk of the tissue, labeling it simply as tumor.
Yes.
So to the human eye, it's just one big monolithic danger zone.
But STORM, analyzing the exact same tissue, saw a completely different landscape.
What did it see?
It carved that single tumor region into 3 distinct separate subdomains. And when the researchers looked closely at the genetic markers STORM identified in domain 3 specifically, they found a hyper-aggressive microneighborhood.
What was actually happening inside that specific domain? Like, why was it so aggressive?
Well, STORM detected that the cells in domain 3 had upregulated specific genes. Things like FEBP7 and RARES2.
Which do what?
They indicate the cells were hoarding lipids, storing up massive amounts of fat to use as energy so they could survive and grow rapidly.
Oh, wow.
It also saw that the cells were adapting to hypoxia, meaning they were thriving even when starved of oxygen. And it detected intense inflammatory signaling.
So it's like a super tumor inside the tumor.
Exactly. It was a deeply entrenched, highly aggressive fortress hiding within the larger mass.
Wait, let me stop you there.
Sure.
If the human expert just saw one big tumor and the AI says, no, there's a highly aggressive microneighborhood right here in domain 3, How do the researchers actually prove the AI isn't hallucinating?
That is the critical question.
Because we hear about AI confidently making things up all the time, right?
And we do, and that is the ultimate burden of proof here. To show the AI wasn't hallucinating, the researchers took the specific aggressive genes that Storm found in domain 3, and they checked them against a massive external database.
External.
Yes, the 𝗬𝗏𝘄𝑚𝘃 database. It tracks real kidney cancer patients and their long-term survival outcomes.
Wait, so they didn't even use their own lab data to prove it. They went out and found a completely independent list of real patients to see if the AI was right.
They had to. That's really the only way to prove real-world validity. And the results were just stunning.
What did they find?
They found that real-world patients who had high expression of those specific domain 3 genes had significantly shorter progression-free survival times.
Oh, man.
Their cancer returned faster, and their prognosis was markedly poorer. So, if we connect this to the bigger picture, Storm is finding microscopic danger zones that human eyes literally cannot see, and those zones dictate whether a patient lives or dies.
That is staggering. It's like finding the needle in the haystack when the human expert didn't even know they were looking at a haystack.
Right.
But here's where it gets really interesting. Finding these hidden danger zones using that expensive, complex ST data is amazing. But the researchers' ultimate goal was to do this without the expensive data. Really.
Exactly. And that brings us to Storm's true magic trick, a process they call virtual ST.
Virtual ST. Okay, how does that work?
Because the model learned the rules of biology so intimately during its pre-training, you can now hand it a basic, standard H&E histology image.
The cheap pink and purple slide.
Right, the cheap one with absolutely no molecular data attached. And Storm can accurately predict the spatial gene expression across the entire tissue.
So it's like, okay, it's as if the AI can look at the physical shape of a speaker cone and tell you exactly what song is currently playing through it.
Yes.
It's seeing the physical shape of the cell and just knowing the genetic software running inside.
That's a fantastic analogy. And to prove just how good Storm is at this magic trick, they benchmarked it against 13 other state-of-the-art models across 11 different tumor types.
How did it do?
Storm completely dominated them. It consistently achieved the highest accuracy, and it's incredibly adaptable. It works on rare, unseen cancers like diffuse large B-cell lymphoma or DLBCL, where other models just completely failed.
DLBCL, so it works even on things it hasn't deeply studied.
Exactly. And it works across different high-tech platforms too, like Visium HD and Xenium. It's incredibly versatile.
So if it can do this virtual ST, what is it actually looking for? Is it just giving a general danger score, or can it find specific things that oncologists actually need to know before they prescribe drugs?
It can find the specific targets doctors need. Let's look at a really memorable example, breast cancer.
Okay.
When a patient is diagnosed with breast cancer, one of the first things doctors look for is a protein receptor called HAR2, or 𝗬𝗏𝗏2.
Right, I've heard of that.
If a tumor is HR2 positive, it means the cancer is aggressive. But it also means the patient can be treated with highly effective targeted therapies that specifically attack that receptor. It also found similar biomarkers beautifully in other cancers, like FGFR3 in bladder cancer.
And normally, finding out if a tumor has HR2 requires a specialized, expensive chemical test in a lab, right?
It does. It requires specialized reagents, trained technicians, and, time. But Storm successfully mapped out the HR2 expression just by looking at the physical shape of the cells on the basic H&E slide.
Okay, the implication there is massive. You're saying a doctor in a small rural clinic who only has a standard microscope and a cheap dye could take a biopsy, run a picture of it through this AI, and potentially spot HR2 breast cancer instantly.
Instantly, without waiting weeks for expensive lab results.
That is insane.
It is the ultimate promise of this technology because it democratizes precision medicine.
It really does.
Right now, getting a deep genetic map of a tumor is just unscalable. But with Storm, you don't need million-dollar lab equipment for every patient anymore. You can take a standard, cheap biopsy image, feed it to Storm, and get a highly accurate map of the patient's genetic tumor landscape.
That completely changes the landscape of access to care. But let's bring this all the way down to the clinic floor.
Okay.
Because standing next to a patient's bed, there's really only one question that matters. Does this information actually help doctors save lives? Did they test if Storm's predictions actually correlate to patient survival?
They did, and they tested it on a huge scale. They applied Storm to predict clinical prognosis using 23 independent real-world cohorts.
23 cohorts?
Yes, comprising 7,245 patients.
Over 7,000 real patients. How did they set up that test?
They took the patient's routine H&E images, had Storm generate the virtual genetic data for them, and then integrated both the physical image and the virtual genetic data to predict how long the patient would survive. Storm dramatically outperformed models that just used the images. And critically, it outperformed traditional clinical staging methods.
Let's talk about what Storm was actually seeing when it predicted if a patient was going to survive or not. What did the high-risk versus low-risk patients actually look like to the AI?
Well, when Storm flagged a region as high risk, it found those areas were densely packed with cancer cells, obviously, but also packed with fibroblasts.
Fibroblasts?
Yeah, supportive cells. Basically, the AI could see the tumor physically building its own scaffolding to spread.
And the low-risk patients, what was different about their tissues?
In the low-risk patients, STORM identified strong immune infiltration. It saw regions swarming with immune cells like lymphocytes and plasma cells.
Okay.
And genetically, it predicted the activation of specific protective pathways like the interferon gamma response. It could literally see the patient's immune system actively fighting and containing the tumor.
But I have to push back here on the practical application, though.
Okay, go ahead.
Doctors already classify cancers by stage, right? Stage 1, Stage 2, Stage 3. If you or a loved one has ever been through cancer staging, you know the anxiety of waiting for that number. Doesn't that stage already tell the oncologist how aggressive the cancer is and exactly what to do?
You would think so, but clinical staging is an imperfect science. There is agonizing uncertainty hidden within those stages. And the researchers highlighted a perfect example in the paper involving colorectal cancer, specifically stages 2 and 3.
What's the clinical dilemma there?
In stage 3 colorectal cancer, the standard medical protocol dictates that everyone gets chemotherapy.
Right.
But doctors don't actually know how long to give it to an individual patient without causing severe toxic side effects. And in stage 2, the decision is even harder. It's highly debated if the patient needs toxic chemotherapy at all.
So you have a situation where some patients might be suffering through the poison of chemotherapy when they don't actually need it.
Exactly.
While others might not be getting aggressive enough treatment to save their lives. And the current staging system just can't tell the difference.
Staging alone cannot separate those patients. But STORM can look within those exact same stages and cleanly stratify them.
Really?
Yes. The research showed that STORM could take a group of stage 3 colorectal patients and separate them into a high-risk group and a low-risk group.
And what was the survival difference?
It was significant. This raises an incredibly important question for the clinic. Could we spare low-risk patients the agony of toxic chemotherapy while aggressively saving the lives of high-risk patients who would have otherwise slipped through the cracks?
That is just incredible. To summarize the journey we've been on today, we started with the frustration of modern diagnostics, right? Where doctors are either flying blind with cheap century-old dyes or completely priced out of the deep genetic truths of spatial transcriptomics.
Exactly.
Then we explored STORM, an AI that was forced to learn the complex rules of human biology by playing biological Sudoku across 1.2 million tissue spots.
Which is still an unbelievable scale.
It is. And we saw how it can uncover deadly, hidden micro-neighborhoods inside tumors that human experts miss entirely.
Like domain 3.
Exactly. And finally, we saw how it can look at a cheap 𝗶𝗏𝗆 slide, see the subtle wear and tear of the cells, and predict a patient's genetic landscape to redefine who actually needs chemotherapy.
It is a profound shift in how we understand disease. We are moving from a medical world where we just look at the shapes of cells to a world where we can seamlessly read the genetic stories those shapes are telling us.
Thank you so much for joining us on this deep dive into the STORM model. But before we go, I want to leave you with one final provocative thought to mull over.
Let's hear it.
If AI can accurately infer our deepest molecular and genetic secrets just from the physical shape and arrangement of our cells in a basic, everyday photograph, how long will it be before virtual biopsies entirely replace physical ones?
That's a massive question.
Right. And if our basic cellular structures broadcast our genetic destiny so clearly to anyone with the right algorithm, what does that eventually mean for the future of our biological privacy? Think about it. See you next time.